our first talk will be from Vincenzo D'Angelella, who is an associate professor at the Oxford Institute for Radiation Oncology. There, his research group focuses on understanding the role of the ubiquitin system in cancer pathogenesis in response to treatment with chemo and radiotherapy. Thus far, studies from his laboratory have uncovered the mechanism of action and function of FBXL17 in medulloblastoma and highlighted a novel synthetic lethal interaction between cyclin F loss and CHK1 kinase inhibition. More recently, his lab has identified the mutations occurring in the E3 ligase KBTBD4 in medulloblastoma that change the substrate specificity and drive the recognition of the chromatin remodeling complex CoREST. Vincenzo has a medical degree from the University of Naples and completed his PhD in the same university in the field of general pathology. Following completion of these studies, he worked as a postdoctoral fellow here in the New York University School of Medicine in the laboratory of Professor Michele Vigano. And while doing his postdoctoral studies, he elucidated the mechanism of action and function of cyclin F, the founding member of FBOX proteins. So today he's going to tell us a bit about his work on neomorphic mutations in E3 ubiquitin ligases. And with that, I will hand it over to you, Vincenzo.
All right, thank you, Brianna, for this very nice and comprehensive introduction. And I would like to take this opportunity to thank the organizer for inviting me, and especially congratulate them for a fantastic series that on a beautiful field, which is growing really nicely. And today I would like to tell you about our story on neomorphic mutation. And I hope I can convince you that this is something that is occurring in E3 ubiquitin ligases, because our classical view has always been, and to this audience, I don't think I have to give a big introduction about this, that E3 ubiquitin ligases, especially of the Kallin family, they assemble these multi-subunit complexes to allow the recruitment of specific substrates. And the presence of a central Kallin scaffold allow to recruit from one side the ring finger protein to recruit D2, and from the other side an adapter for substrate specificity. And there is an important modification here of ubiquitin-like molecule, NED8, that allow the transfer of ubiquitin molecule to the substrate. And we have seen over the past few years the growth of a field that has been quite amazing to see. And the fact that there are small molecule and PROTAC that they can hijack adapters in order to allow the recognition of neosubstrate. But today I would like to tell you about our finding, and I hope I can convince you that there is another possibility beyond chemicals to allow the recruitment of neosubstrate. And this is that mutation in adapters can change the substrate specificity to allow the recruitment of neosubstrates. And actually, my lab, as Brianna has mentioned, has been working on characterizing E3 ligase substrate pair for a while. And I am an MD by training, so I always like to do research in a disease context. And especially, we worked on brain tumors. We have summarized the knowledge in glioblastoma and medulloblastoma for E3 ubiquitin ligases, and there are a lot of opportunity there. So it's a very exciting field. And my interest in these findings stemmed from my interest in medulloblastoma. And medulloblastoma is quite a bad tumor. It occurs in children. It's the most common malignant childhood brain tumors. And unfortunately, there are not a really good set of therapy, and we can distinguish now 4 subtypes based on their transcriptional profiles, and which correspond also to the pathogenetic mechanism at the basis of the disease. And we can distinguish a Wnt subgroup in which there is hyperactivation of Wnt signaling that allows the increased development of progenitors that then populate and become medulloblastoma. There is another group in which they called sonic hedgehog, in which the characteristic is hyperactivation of sonic hedgehog signaling. And then we have a group 3 and group 4 for which we call them group 3 and group 4, because essentially we don't know what are the pathogenetic mechanism at the basis of this disease. And unfortunately, these are the groups of kids that they also do the worst, because they have a very poor prognosis. And also, even if you treat a kid with radiotherapy and chemotherapy, which are conventional therapy that they work more or less, you know, like with a certain efficacy, the kids, they will develop long-term sequelae. So we need to identify targeted therapy, novel target, especially in kids, to avoid to give them bad, you know, unspecific treatments. And one effort that has been done from this point of view was a great effort from the Paul Northcott lab and Michael Taylor, that they've classified the most frequent gene that they are mutated in medulloblastoma. This was a paper that was done in 2017, and among the top 10 most frequently mutated gene in medulloblastoma, there was a gene called KBTBD4. And this was, for me, quite an exciting finding, because it was obviously an E3 ubiquitin ligase of the CAL3 family. And as you may well know, the characteristic of these E3 ubiquitin ligases is that they have a BTB domain, which recruits a CAL3 complex, and which in turn recruits a ring finger containing protein for the E2. And then they have a Kelch domain, and this Kelch domain is required to recruit substrate. And the mutation in KBTBD4, they were quite interesting, because they were all residing within a particular region of the Kelch domain. So although most frequently in E3 ubiquitin ligase we have inactivating mutation, this didn't look like an inactivating mutation. It looked more like a gain of function mutation. However, my good friend and collaborator, structural biologist Alex Bullock, had a different idea. He thought that actually these mutations, they were very different from each other. So what they did, they kind of changed the surface of the Kelch domain by pushing this residue isoleucine 310 within the Kelch substrate. And he was thinking maybe they are neomorphic, so they allow the accommodation or the recruitment of a different substrate within the Kelch domain. And I have to say, I was quite skeptical about this, but we did our work to identify substrates. We have done this for a number of protein, and it worked quite well. The approach is quite simple. We transfect protein, we overexpressed the adapter, and we treat cells with a compound, MLN4924, which blocks the net deletion of this complex, so allows the trapping of the substrate on this complex, because it prevents their activity on the substrate, their ubiquitination activity. And we did mass spectrometry to identify the interacting partner. And we compared KBTBD4 wild type to the two mutants. And as you can see here, the KBTBD4 wild type and the mutant, they all bind CAL3 and RBX1, which was expected because they are, it's an E3 ubiquitin ligase. But the surprising thing that we identified was that two mutants, they are very different in their composition of amino acid. They both bound to a particular complex that is called CoREST. And this is, we did not only isolate the CoREST per se, but the entire chromatin remodeling complex was coming down. So we got CoREST, LSD1, GSC1, HMG20A and B, but also HDAC, which is another component of the complex, which was quite nice because we could say two different mutants, they were doing the same thing, and this was obviously not by chance. And we confirmed this interaction by simply immunoprecipitation. In the medulloblastoma cancer cell line, in these cells, we can overexpress KBTBD4, and we can isolate the CoREST complex together with LSD1, all in the presence of MLN4924, which suggests, and only when in the presence of the mutant, there was no interaction between the wild type and CoREST. This was indicating that actually the KBTBD4 mutant was able to degrade CoREST, because we could only identify the interaction in the presence of MLN 4924. So to confirm this finding, we actually generated a cell line in which we can control the expression of KBTBD4 using a doxycycline inducible promoter. And in this case, we used increasing concentration of doxycycline, in which using this increasing concentration, we can control the expression of KBTBD4 wild type. And as you can see here, in the red square, there is no degradation of CoREST when we express the KBTBD4 wild type. Instead, when we express the KBTBD4 mutant, even at a lower level than the wild type, we could observe a very striking degradation of CoREST or disappearance of CoREST. And this we know that is degradation because we could rescue it by proteasome inhibitors or MLN 4924. And the same happens with another component of the complex, LSD1. So what we did, we actually wanted to confirm that this was ubiquitination followed by degradation. So we used tandem ubiquitin binding entities that we generated in the lab using ubiquitin as a ubiquitin binder. So it's important to mention that these chain binders, they are completely chain agnostic, so they can isolate every ubiquitin chain present in the cells. And as you can see here, the result, they were quite striking. I was really surprised by these. I've never seen a tube like this. So, and this is because the KBTBD4 wild type had no effect essentially on the CoREST ubiquitination. Instead, when you express the two mutant, there is a reduction of CoREST, which corresponds to a ubiquitination of the CoREST protein, which was quite nice to see. So what does CoREST do? Why this is important in these tumorigenesis settings? And CoREST is a transcriptional repressor that normally keeps transcription at bay by demethylating H3K4 in order to silence chromatin. So the hypothesis is that the mutant KBTBD4 is actually acting as a Pac-Man and is actually chewing up CoREST in order to induce the H3K4 hypermethylation and activating transcription. And this was exactly what we observed. We observed that when we express the two mutant, we observe an increase of H3K4 dimethylation. So we asked, can we also activate transcription using the same approach? So we use D283 cell line, medulloblastoma cancer cell line, in which we can express KBTB4 under doxycycline inducible promoter. And as you can see here, we can see KBTB4 wild type, but there is no effect on transcription. Instead, when we express the mutant, the situation was completely different. And this was quite nice to see because, first of all, we observed that most of the genes, they are transcriptionally activated and they're not repressed, which means that there is an effect on a transcriptional repressor and not on a transcriptional activator. And second, we could observe a number of CoREST targets. This was very nice. It confirmed our hypothesis. But when we looked at the gene enrichment to understand what were the set of genes that they were upregulated, there was a little bit of surprise because we observed a lot of pro-neurogenic program, genes involved in axon guidance, glutamatergic synapse, which was quite counterintuitive because why would you have an upregulation of pro-neurogenic signaling when you want to promote cancer? And there was also no effect on proliferation because when we express this KBTBD4 mutant in our cell line, we don't have any effect in increasing cell proliferation. So how does KBTBD4 induce tumorigenesis? Because this is the sole genetic driver in a subset of medulloblastoma. And some of the clue came from observation from the lab of Gail Monaghan that showed already in 2016 that mouse knockout for CoREST, they have an interesting phenotype. They die at birth, but their brain, as while in a normal situation, you have an expansion of the striatum, and the majority of these cells, these are neuron, although you cannot appreciate at this low resolution. In the subventricular zone and in the ventricular zone, you have progenitors. And instead, when you remove CoREST, you have a large expansion of progenitors here, where there is the asterisk. So it almost looks like that when you remove CoREST, some neuronal progenitor, they are becoming more proliferative and they have an increased stemness capacity. So we looked in our data set if there were an overrepresentation of OCT4 nanogase OX2 target, which are stemness-like signatures. And we observed exactly that there was an increase of the stemness gene. And so to prove that in vitro and in cell that actually there was an increased stemness when we overexpressed our KBDPD4 mutant, when we expressed our gene, we actually did two assays. We first measured the capacity of these cells to grow in an anchorage-independent growth, so the capacity to form medullospheres. And as you can appreciate here, the wild type doesn't have any effect. Instead, the mutant KBDPD4 has a striking effect in increasing the formation of medullosphere. So it looks like that these cells, they are more able to grow without attachment, and which is a characteristic of stem cell. Another characteristic of stem cell is that the cancer stem cell, they have an increase in aldehyde dehydrogenase activity. And this is very easy to measure because there is a model substrate that can be converted and used for FACS analysis. And the model substrate turns cell red, and you can easily see it by FACS. And when we express the KBDPD4 wild type, the number of cell expressing aldehyde dehydrogenase activity was only 12%. And when we express the mutant, we observe a striking increase in the number of red cells. And this is, I think, is a representation of stemness activity of these cells. And it's interesting also to note that when we use LSD1 inhibitor, we don't have a further increase above the baseline of KBDPD4 mutant, indicating that KBDPD4 mutant is almost getting rid of all the CoREST complex that they are associated with LSD1 in cell. And then finally, we needed to do obviously some mouse models to actually prove that this was really effective. But unfortunately, the majority of these experiments, they were done during the pandemic. And as you can imagine, it was difficult to keep mouse alive. So basically, we went directly into human models. So we collaborated with Steve Clifford in Newcastle, and he has a cohort of about 250 medulloblastoma cases in which we compare the transcriptional signature that we have in our cell line to the transcriptional signature present in medulloblastomas. What we could identify was the presence of 3 clusters. And among these 3 clusters, there were 2 clusters, which we call cluster 2A, cluster 2B, which showed a very nice upregulation of CoREST target, a very nice upregulation of, which corresponded to the gene upregulated in our KBDPD4 mutant. I have to say that some of these tumors, they have KBDPD4 mutation. So all the cluster they encompass also the KBDPD4 mutation, but there is a very large subset of tumors that have a similar signature, but they don't have the mutation. And this corresponded also to a signature of stemness, because we observed that there is an increase of nanog-and OX4 as OX2 target. So I think our final model is that during normal cerebellar development, KBDPD4 is doing something else, which we don't know exactly what is it yet. And the CoREST is able to exert this activity of transcriptional repressor to allow the differentiation of progenitors that then they will populate the cerebellum. Instead, when there are neomorphic mutation in KBDPD4, this leads to the degradation of CoREST and induces the activation of the transcription of a set of genes that they are stemness related, that would favor the proliferation of cellular progenitors that then they will induce the development of medulloblastomas. So I think I would like to summarize a little bit what I've shown you so far. And the fact is that we have identified KBDPD4 mutation that elicit neomorphic cognition of CoREST, and these drive tumorigenesis, we think through stemness. I think there is also another implication for our finding is that human medulloblastoma group 3-4 present CoREST alteration that they are well beyond the mutation, which could be a novel pathogenetic mechanism. I've also shown you that E3 ligase can mutagenize to divert their substrate specificity. And this is, to my knowledge, the first example of mutagenesis leading to difference in substrate specificity. And I think if this is the only example to my knowledge, but if there are other examples, I think we have a new approach here. We could target this mutation with PROTAC and other small molecule to generate first-in-class targeted products, which creates opportunity for cancer or other diseases in which there are mutations in E3 ubiquitin ligases. And I have to mention that there is a small molecule that does exactly the same of what we showed with the mutation. This is called UM171, and we have heard, I think, a brilliant talk by Subramanian in this series about UM171. And UM171 is a molecule that is used in phase 2 to drive stem cell expansion, and it does exactly the same as the mutation that we have identified. It engages CoREST to drive its degradation. So thinking a little bit about this process and the analogy between this small molecule and mutation, I came up with an idea that I hope it stimulates a little bit of thought in the field. And the fact that I hypothesize that most probably E3 ligase have a limited available degradative space. And this is normally restricted by the presence of the degron, so that each ligase A can only target a substrate B, C, or D, according to the degron. We can expand this available degradative space with products, and these is not only molecule, but is also peptides, and these could be infinite, this expansion in theory. But I think that for molecular glues and other small molecules, the space, the available degradative space is quite restricted. And I think there we would have to start designing with taking into account the biophysical property of interaction and the composition of the degron. And most probably one idea is to use mutation to probe this available degradative space to actually identify the potential substrate that we can hijack. And I would like to thank all the people that they contributed to this work. Without them, this would not be possible. And especially the two people that they were the leader in this project, Raphael, Joris, and Zhuo Yao Chen, and my entire group, which I'm very proud to have, as you can see from my face in the picture here. And I would like to thank also my structural biology collaborator, Alex Bullock, and the proteomic that we have and collaborator, Benedict Kessler, the bioinformatics that help us to define the transcriptional signature, Francesca Baffa, Luca Busino at UPenn, Ava Van Essen, especially Newcastle University with Steve Clifford and Stacy Richardson, that allowed us access to a number of medulloblastoma cases in which we could confirm some of our findings. And finally, I would like to show you some of the Oxford activities in the rowing that we do. And I'm happy to receive any question.